Okay, great. We can get started. Thank you for joining us, everybody. My name's Sarah Barton. I am the Strategy and Operations Manager for the Research Platforms team, which is the team that supports the Sustainable Futures platform. This is a new platform at the University of Manchester based around environmental sustainability, which has been developed over the last 18 months. And we've had a number of large information gathering events over the last year, followed by the appointment of Mike Shaver as director on the 1st of November. So next up, our second speaker is Dr. Neil Dixon, who's also from the Manchester Institute of Biotechnology in the Faculty of Science and Engineering. So I'll hand over to Neil.
Okay then, are we all good?
Yeah, perfect.
Excellent, thanks Sarah. Okay, and thanks for a very nice introduction. So my name's Neil Dixon. I'm based in the MIB. I guess kind of back to earth after Alad's very nice talk. I'm going to talk to you about some of the global challenges that exist and some of their biological origins and how potentially biotechnology can have a role in addressing some of those problems. So a huge number of the challenges we see on planet Earth are actually kind of have a political origin and could be addressed by political issues. For example, as we look at global inequality, poor access to healthcare, to good water, sanitation and finance are some of the driving issues that lead to other politically related issues such as mass migration. If we look on the other side, there's a whole series of biological related challenges. And indeed, as we said at the outset, it could potentially have biotechnologically related solutions. Obviously, global pandemics have a biological underpinning and solutions have been met through biotechnology. If we look at ecosystem damage, biotechnology has a role in trying to limit the release of toxins or xenobiotics into the environment, and advanced forms of biotechnology, environmental biotechnology, could provide potential solutions to remediate damaged ecosystems around the world. Similarly, around climate change, if we can develop technologies that make fuels and chemicals more sustainably, we can think about reducing our impact and our dependence upon fossil fuels, reducing CO2 emissions. And similarly, we can think about engineering organisms and microorganisms to mitigate some of that already released carbon dioxide. So we can think about CO2 fixation. Related to that, finally, then in the food area, we can also think about engineering soil bacteria in microbiomes to facilitate better nitrogen fixation and plant growth. So if we look about greenhouse emissions, we can see a huge swathe of those come from energy, a large amount actually just heating in buildings and transportation, but a huge around quarter of all the energy, all the greenhouse emissions come from industry use. So industry often has a double whammy. So not only is there greenhouse emissions associated with the energy required for that particular sector, but also the manufacture process, for example, in chemicals actually lead to greenhouse emissions themselves. So see, these are some of the challenges and potentially areas of focus as we try to reduce our CO2 emissions. So I'm going to show you this biomass of life chart here, a really nice visual graphic that shows all the sources of carbon in the world and how they all relate. And you can see down here, this orange block here, which is animals. So that's all animals, all humans, all wild life, all livestock, equates to around 4 gigatons of carbon. That's a huge amount of carbon on the Earth. But if we compare that to the amount of plastic that we've made over the last 100 years or so since we've been making plastic, unbelievably, we've made twice as much plastic as there is biomass in the whole of the animal kingdom. That's just an outstanding amount. And you think this is essentially a waste material, pretty much single use, less than a single percentage of plastic is recycled. So a huge amount of this goes to either incineration, goes to landfill, or ends up into the environment, into rivers and waterways. So it's really not very good for our planet. So we need to try to find ways to better deal with this waste and potentially convert the carbon that's within that plastic into a resource, a feedstock, if you will, so we can find cyclical uses of that carbon and to fuel biotechnological processes. If we compare the amount of biomass to that in bacteria, so bacteria are found everywhere throughout the environment, in your gut, all over the Earth's crust, and they're widespread. They live in lots of different environmental niches, and the total bacterial biomass on Earth is around 70 gigatons. So that's a huge kind of resource, if you will. They're widespread. Bacteria, because they live in so many different environments, they're a good source to go and find new enzymes that carry out exotic biotransformations that could deal with, for example, some of this waste material we put in the environment. But also the bacteria themselves are a particularly useful source, as they can live in so many different environments. They could be used as hosts to function in different extreme environments. So, and the final slice of that tower I'll just expand upon is plants. We look at the huge amount of carbon that's locked away in plants. That's 450 gigatons of carbon. That's a huge renewable source of carbon that could be used more effectively and could be used to avoid our reliance and dependence upon fossil fuels. So what are we doing in this space, and what could we do? So if we look around the way that we classically use biomass to make fuels, we can look at first generation biorefinery, where bioethanol is made through the saccharification and fermentation of sugars that are derived from sugarcane or from corn syrup or from wheat. And that is then broken down and fermented to produce bioethanol. Certain countries around the world sell their petroleum or their petroleum ethanol blends at very high blends, for example, around 8% bioethanol. The most common biopolymer on Earth is cellulose. So this can, in principle, be also processed by so-called second generation or 2G biorefinery processes, again, to make fuels, bioethanol. This is kind of borderline economically feasible. There's only a couple of plants around the world really operating at scale and doing this. But it shows you the potential, at least, of making an advanced biorefinery and using a very common carbon or biopolymer source and converting that into a valuable product. As we move forward, there's a lot of interest certainly in the research domain over the last 10 years of taking lignin. So lignin is a complex recalcitrant material. It gives plants its main structural integrity. It's the second most abundant biopolymer on Earth. It's very heterogeneous, so it's quite difficult to process. But in principle, one can break that down into its individual constituent components, and then those compounds, those monomers, can be funneled down through degradation pathways, through biological catabolic pathways into central metabolism, and the energy from that can be then used to drive biosynthetic pathways to make a range of chemicals, materials, and fuels. As we move on and think about things beyond just kind of biomass as a feedstock, we can think about in the future using plastics, using the carbon within plastics and potentially other types of xenobiotics in the environment, and using these, breaking these down with enzymes into their individual monomers, and using the carbon in those monomers to drive microbial metabolism, which then we can use to produce various compounds of utility for society. Where these two kind of biomass and plastic areas coalesce is because there's a lot of similar catabolic processes. If we look at what are called the upper aromatic degradation pathways in nature, these are the kind of compounds, the aromatic compounds you get from biomass derived from lignin. Once those are broken down, this goes to a key intermediate compound here called Protocatechule, or PCA. That is then cleaved and goes into the central metabolism. The citric acid cycle allows energy to be produced from that particular carbon source. If we look about the aromatic degradation, particularly for this compound terphthalate, so that's one of the building blocks that's found in polyethylene terephthalate. That's the plastic that's made in all our drinking bottles we hear so much about. The way that's broken down, that TPA, goes through a common intermediate, this PCA compound again, before it goes into central metabolism. So this is a kind of an overview of some of the areas I'm going to talk to you about over the next few slides. We've developed means to try to add value, convert this compound here called ferulic acid, and convert that into higher value compounds. We've also developed biosensors, those ways that we can detect and sense for the presence of this compound. Similarly, we've also detected biosensors so we can detect this key intermediate compound here, PCA, and also this plastic breakdown terephthalate. And we're starting to work on pathways where we can do breakdown of this TPA compound into central metabolism and make higher value products. Okay, so into the talk proper. We'll initially talk about some ways we've tried to add value to this lignin cellulose. So this is a chemical structural representation of lignin, and it contains a range of sugars and these aromatics. If we can cleave this ester bond and then we release an acid, we can carry out a double chemical reduction and make this coniferol compound here. So this is a flavor and fragrance compound. It's highly valuable, but also it's a key intermediate to a range of other chemicals. So we can make other flavors and fragrance compounds and a range of active pharmaceutical ingredients, as you can see around here, antiviral, anti-cancer agents, and so forth. So it's a logical pathway to try and make this chemical more sustainably. So the way we've done this is through a kind of hybrid enzymatic whole cell biotransformation process, where we initially take a biomass. In this case, we take a wheat straw, treated that with an enzyme, and then from the end products from that, we then carry out a biotransformation. These are two enzymes that we incorporate into an engineered bacteria that allow the ferulic acid that we can show here to be reduced down all the way to this primary alcohol product, this coniferol compound, our target compound of interest. So we reported this a few years ago. We compared the use of two different biomasses, a wheat straw and a wax, and both using an enzyme and an alkaline hydrolysis, and the enzymes work very well, giving us good quantitative release of the acid from that biomass. We then incorporate the whole cell biotransformation process, and we can make stoichiometric amounts of our target compounds. What's kind of interesting is the value of the starting material versus the value of the product. So as you factor in process costs, that whole change of value added reduces down, but at current, it's around a 70-fold increase in value between starting material and product. As you increase the efficiency of that process, obviously that fold change would increase. So that's just a demonstration of how you can create value from essentially what is a waste agro-industrial residue. They kind of burn these residues, so they're very low value applications. So that's kind of the biocatalysis that we've done. We're going to move now towards some of the biosensing types of technologies. So as we've just heard, we can convert some of this waste agro-industrial residue into these higher value materials. But as we engineer microorganisms to do more bespoke transformations, we need to develop tools and technologies, both so that we can monitor how efficient that conversion is occurring, but also how we can regulate those pathways inside those cells. So that's why we develop these things we call whole cell or genetically encoded biosensors. This is just a schematic representation of what we try to develop. If you think about breaking down biomass into its individual components, you will release a series of these aromatic compounds and sugars, and then you develop this genetically encoded biosensor, which is essentially a genetic circuitry that we put inside a bacterial cell that allows you to transduce a chemical signal into a gene expression output. And if that gene output is a fluorescent protein, we can then monitor in real time the process efficiency of how this biomass is broken down into these individual components. We could also use this for screening new enzyme variants, for example, doing direct evolution on different enzymes. And similarly, we could link this output gene to the expression of another gene in the catabolic pathway. So we could use this for flux optimization through an engineered biosynthetic pathway. Okay, so that's the concept. What have we done in this space? we developed a series of different biosensors. First off, this key compound that I talked to you about, ferulic acid. It's derived from biomass. It can be used to make vanillin and other high-value compounds, as well as coniferol I just talked to you about. The biosensor has 3 different components here: a reporter gene, a regulator, and an activator gene. But essentially, the take-home message is, by using this, we can use this to sense for the presence of these aromatic compounds that are derived from biomass, using the fluorescent protein readout here on this x-axis with these different homologs of ferulic acid shown here. To use this in anger, what we've done is screen 3 different types of biomass with different enzymes. We carry out enzyme treatment on these and take the supernatant from that enzyme pre-treatment, and then incubate that with our biosensor, and then the fluorescence readout we get from our biosensor, we can use then to screen for enzyme activity and indeed for the composition of that particular biomass. So the second biosensor that we developed is to this key intermediate compound I talked to you about between these upper and lower pathways, this TCA compound here. This is a much simpler biosensor, just 2 components, the regulator and an output gene. It works with a much greater narrow specificity, but gives a much better signal over around 100-fold. So it's a very active biosensor and responds to these 3 compounds. What we were interested in doing is saying, can we perform protein engineering on this particular transcription factor to change its ligand recognition potential? So natively, it responds to this PCA compound here, which you can see in this crystal structure. If we can mutagenize around this ligand binding site here, can we change its ligand preferences? So we've done that, and we've mutated it, and through a series of screenings, we've identified that the mutants no longer respond to the cognate ligand shown here, but indeed do now respond to and activated by a range of aromatic aldehydes, including vanillin. So vanillin is a high-value compound. It's an analog, or it's one of the main components of vanillin, which you extract from vanilla pod, which is particularly limited and quite expensive. So there's a lot of interest in being able to make these flavors and fragrances via fermentation, via biotechnological routes. So having a biosensor for that process would be particularly useful. Okay, so shifting away next from plant biomass, I'm going to talk to you now around plastics and the work that others are doing in this space and that we're starting to work on as well. So around 6 years ago now, a group in Japan identified this bacteria called Iodinella saccharinitis that was able to grow on PET plastic, so polyethylene terephthalate, this low crystalline PET film they used. And the two key enzymes that the organism encodes are these 2 hydrolytic enzymes here called a PETase and an MHETase. They cleave the pristine PET and form this intermediate MHET, and the second enzyme cleaves that into the constituent components, the 2 monomers of TPA plastic, which are ethylene glycol and terephthalic acid, or TPA. So prior to that, another group in Japan around 10 years ago had studied this organism, this Kumamona, that could take TPA and could catabolize that, it could break it down. So it does a dioxygenation initially, breaks that down to the DCD intermediate, and then that is further broken down into this PCA, this key intermediate compound, which is further cleaved and broken down into central metabolism. So there's lots of work going on the back of this science paper on trying to use these approaches. Lots of researchers have engineered hydrolytic enzymes, PETases and MHETases, that work at higher temperatures, so work above the glass transition temperature of PET plastic. We've been particularly interested in the structure of the catabolic genes, and particularly the operon that encodes these genes. And specifically the transporter, this TPHC gene here, and this regulatory protein here called TPHR. And the next couple of slides are going to unpack some of the work we've done in that space. So the TPHC gene, the question is, how does TPA get inside a bacterial cell? So here it's shown as a protonated diacid, but actually because of the pKa, the ionization state of that TPA, at neutral pH, that would be doubly negatively charged. So it wouldn't easily passage across a lipophilic cellular membrane. So the cell needs to use active transport to get that inside the cell.
It uses this using this TPHC, which is a solute binding protein. It binds to it. This is believed how it works. Then that delivers that substrate to the membrane. It's transported across, and then catabolism can occur. So we wanted to study this TPHC protein. So we used a range of physical and biochemical techniques to study its ligand interactions. So we crystallized this protein in both open and closed forms. That's closed form here, shown in the yellow, with a ligand bound. You can see this structure here, this particular alpha helices are moved upon this ligand binding. So this is a conformational change. Essentially, once this ligand bounds, that allows this then to be released. If we zoom in on the active site, we can also see the particular amino acids that the protein uses to recognize this terephthalate compound here in the active site. We've also done some bioinformatic analysis, so we can look at the sequence of these proteins and look for homologs that are in bioinformatic databases. And by doing that, we can then do some gene context analysis. So we can go into those genetic databases, those genomic databases, and look whether these particular solute binding proteins that we're interested in also have flanking genes. So those are genes next to it in the chromosome that also look like they're associated with the breakdown, the catabolism of this TPA compound. So that's what we've annotated on top of this phylogenetic tree here. These are where they are located within loci, genomic loci that look like TPH operons. So that's what we've shown you at the bottom of the slide here. We can see a range of different operon structures. So there does look to be some variation in the sequence homology and indeed the composition and structure of these operons across different taxonomic groups and where we've identified these operons from. Okay, so moving on now to work we've done on the regulators. So we've been talking initially on this transporter, this TPHC gene. We're also interested in this regulator, this TPHR protein here. So we use these parts, which we've mined through this bioinformatic database, and then pulled those out and said, okay, so how are these different homologs functions? Do they all work the same or they work differently? So that is where we've taken these elements in red boxes and then cloned them in front of a reporter gene. So this is just a fluorescent protein, and that allows us then to monitor the performance, the expression level, and indeed the sensing capability for this key breakdown product from plastic. So here's just some proof of principle data. This is quite early data. So essentially, we've cloned in 3 different regulators and promoters. That's DNA sequences upstream from the reporter gene. And then we just monitor the reporter activity expression by increasing that TPA, that active compound. And you can see different performance attributes there. So we think these could be useful then for sensing, for increasing enzyme activity, and for regulating the biosynthetic pathways. Okay, so the final slide, just to kind of maybe put a bit of future perspective of where this goes and what we could or what might want to do in the future. We've talked about plastic biorefinery, where we could put in hydrolyzable plastics and use those in a more sustainable way. So instead of these plastics being incinerated or going to landfill, can we collect that through the municipal waste system and process that carbon and make higher value? We might start to think about some of the solutions for contaminated environments. Obviously, if we're going to harvest that plastic or those waste materials already in the environment, there is a cost associated with that and pulling that back in. So there's a real big question about whether that's feasible economically to go and recover these materials from waste environments and put those through industrial processing. Another way that we might like to think about trying to solve some of these contaminated environments are more in situ solutions. So could we, for example, engineer microbiomes in these communities where these xenobiotics are found? And could we solve some of these issues by engineering those native systems to better break down those waste materials? So you could do this via a classical route. For example, you could isolate those individual bacteria in that microbiome, find out which are the preferential bacteria, and then grow them up and then feed them back in. That would be a kind of a native or bioaugmentation kind of approach, not using genetic engineering or genetic modification strains. The sort of route B is where we would modify them and bump those up through gene editing or some other approach where we would engineer enhanced capabilities and introduce those. The other kind of more advanced application, which is a real kind of emerging application and feeds over to lots of different areas in terms of microbiome engineering, is where we could do in situ delivery of payloads using mobile genetic elements. Could we put useful biosynthetic pathways into those endogenous communities rather than putting non-native bacteria in? Could we just incorporate the genetic material directly into those organisms? So that's kind of just a little snapshot and overview where I think this technology could go in the future. Okay, I'm just going to acknowledge all the people that did this work, various different photos of people from my research group over the years, current members here in blue, past in grey, collaborators and funders, and then say thank you for your attention, and I'm happy to take any questions.
Thank you so much, Neil. And we do have a non-specialist question or a suggestion comment from Maggie, which is around, Maggie has read that PET biodegradation can lead to the formation of biodegradation of PLA or PHA. And she's wondering whether there's an interesting upcycling loop to look at there. Degrade plastic to help produce bioplastic.
Yeah, indeed, you can. There is enzymes that will break down these ester-based things. So PLA can be broken down into individual components with esterases, lactic acid. So you can break them down. So you could make pristine PLA again. You could recycle it back, or indeed you could think about putting that carbon from the PLA into central metabolism and making other things. So there's anything that's kind of hydrolyzable and ester-based is kind of the low-hanging fruit, if you will. That's the kind of easy one. It's the non-ester-based carbon that is going to be the real big challenge in this space.
Thank you. And we do have a couple of other questions. What do you think the major barriers are for industrial scale adoption of using these alternative feedstocks for chemical production?
Yeah, I mean, I guess incumbent costs for chemical production, you're competing against industries that have got locked in processes invested. You know, these are big industries at scale and they're big processing plants. So those processes have been generated over many years and relatively they're generally very efficient. So you've got to be competing against those incumbent technologies. So that's a huge barrier really. And so I think really some of the facilitators of overcoming that, the efficiency competition, I guess some of that is potentially coming through legislation. And I think that can really help. And generally chemical companies are keen to increase their green credentials, both as a PR exercise and in all honesty as well. Maybe the petrochemical companies maybe less. Be careful what I say on public discussion forum. But yeah, I think there is a move across the whole chemical production sector to be perceived and to do the right thing. But certainly legislation will help.
Of course. And what do you think the biggest hurdles of implementing these engineering biology solutions for bioremediation are?
Yeah, I mean, I think that again, I think legislation is going to actually really help. If you think about what happens with waste, what about the stuff going in rather than the stuff that's already out there first. We need to stop investing in incinerators. So there is a big push kind of a waste to energy, and the UK keeps investing in new incinerators. So the idea is that we'll just burn the stuff and we'll generate energy from it. But that's not really going to be achieving the net zero targets that we've committed to as a country. So that's a real big hurdle. If we could if the policy can change around that, I think that would certainly provide some economic pull that needs to be happening. On the more in situ solutions, I think there needs to be, it's around public perception again, around policy and release of genetically modified or genetically edited organisms into the environment. Obviously, currently it's all very restricted, although there is potential where we can start to think whether there's a greater acceptance for certain types of areas. I think plants is always a controversial one, anything to eat. People seem to be against the sort of genetically modified plants. But there is examples of where we are and have released genetically modified materials. Certainly the gene drive technology that's being used to eradicate malaria, to engineer mosquitoes. I think that's a really interesting space in terms of the public perception and that where different technologies could potentially be accepted. So I think there's a public perception and a policy piece around that.
Yeah, so again, the interdisciplinary aspect is key. And thank you, Neil, for a fascinating talk. And thank you to both speakers. So we did have one quick question from Maggie. Is there a mood you think to more readily accept genetically edited plants?
Yeah, so it's interesting. So DEFRA just did, the Department for Food and Energy and Rural Affairs just did an open consultation last year. It's on their website around gene edited technologies. And I'm not going through it in great detail, but it looks like there's a greater acceptance, at least more willingness from the government and from technology developers to promote and facilitate the use of gene edited technologies. That's gene edited with a very narrow focus of what gene edited means. So things that you could already have developed through plant breeding, where you do it by a very selective genetic technology, that seems to be more likely to be acceptable. It's certainly from a regulatory perspective, whether as an individual user that is accepted, that's always a question that's kind of down to personal choice. But there is a PR issue here, yeah, that certain sectors, people who accept genetically modified or genetically engineered products, yeah. So insulin is GM, so is vaccines, they're all GM. So, or majority, certainly all the COVID vaccines are GM. So people have to take things for therapeutic purposes. So I guess there's a PR issue around biotechnology more broadly. I think we should all engage with.
That's a really important point. So a comment from Isaac about the huge amounts of research into recyclable materials rather than reusable materials.
Yeah, I mean, I think all these things are going to go in conjunction, aren't they? What they call it, the 6 R's now, and I'm going to remember all of them. Of plastic reduction and material reduction in use. Certainly recycling and reuse are one of those. So I think there's a provision of a range of solutions, and you never know whether it's going to be one or a combination of those, most likely a combination that's going to provide us with more sustainable futures.